Hi, my name is Barbara Harrington. Today's date is March 23rd, 2018. This week we went over bacterial morphology and staining techniques. So if you come over here, I'll show you what the first part of the lab that I already had performed. It was when we used mouth swabs and applied it to a slide, and then we used heat-fixed technique, which is where we took the slide. Once we took the swabs and we put it on the slide and labeled it, we put it over the flame 5 times, trying not to directly burn the slide, but the purpose of the heat-fixed was to burn off the bacteria and help keep the cells that we want to observe fixed onto the slide. So this to the left right here, this observation is a direct stain where we used our crystal violet onto from our mouth swab. And as you can see, this is what I found under the 40 times magnification, which is a total of 600 magnification. And then to the right here is our mouth indirect slide, which again, I used a mouth swab onto a slide and then used the Congo red. We didn't directly heat fix this one on the right. It was air dried on the right. But as you can see, I did not get as many cells appearing. This is even at the 40 times magnification, which is a total of 600, but I still did not get as clear of a picture as on the left. The second part is our gram staining process. So there's 4 steps to this process. Right now you caught me at, sorry, let me get it in order again. You caught me at step 2, where I'm using the S epidermis that we inoculated from our nutrient broth that we used last week in our slides. So I've just been saving that for the past week, and I'll keep saving it for as long as I need to use it for these next couple weeks, so I can I don't have to keep on culturing these different bacteria, because it takes about 48 to 72 hours to incubate properly. So I already did step 1, which was using the violet staining. I let it sit for 60 seconds after applying the violet staining, and then I rinsed it off. And then the second step is the PVP iodine, which I also let sit for 60 seconds. And then, as you just saw, I rinsed it off with water. This is our third step, the decolorizer. So the decolorizer will work where if it is a gram positive, it will keep the purple stain. If it's a gram negative, it will actually strip the color of the purple. And this is one way that we help decipher between gram positive and negative. The final step, which I need to rinse this off, it's been about 30 seconds. Gently rinse this off into my tray. Here. Okay. Try not to disturb my slide. And then the final step of the gram staining process is our saffron gram stain, which is like a reddish color. So if it's a gram negative bacteria, it will actually keep that red dye, and we'll see it under the slide. It will appear red. Gram positive will appear purple. An example, because I just did the E. coli staining prior to the video. This is my E. coli gram stain. As you can see, it has this pink appearance. So I am going with that this is a gram negative bacteria that we are observing here. I just have these last 2 to do, and then I'll be completed. Thanks for tuning in this week. and catch you next week.